Good morning, everyone, and welcome to the second day of the ABG's Life Science Summit. My name is Gonzalo Artiach, and I'm a biotech equity analyst here at ABG. The first company of the day is Cereno Scientific, a company developing new drugs for cardiovascular diseases. With us, we have here Sten Sorensen, the CEO. Please, Sten, the word is yours.
Thank you, Gonzalez. And thank you for the invitation, ABG. I'm very excited to be here and report on our progress with Cereno Scientific, which has been quite dramatic over the last year. So, Cereno Scientific is developing new drugs for rare and common cardiovascular diseases. It's built on a vision by Professor Sverker Jern to uncouple the potential of epigenetic modulation through HDAC inhibition for patients that suffer of cardiovascular disease, the most common cause of death in the world. Our lead program is CS1. We have that in phase 2 in PAH performed in the US. We have, in addition, 2 preclinical programs that we're performing in a collaboration with the University of Michigan in Ann Arbor, Michigan. So we have a headquarters in Gothenburg. We have an affiliate in Boston, Cereno Inc., and major R&D projects in the US, as I mentioned. This vision of Professor Sverker Jern and the drive that we have and passion for these developments have been attracted a strong following from thought leaders, global thought leaders around the world. I have myself pursued 2 paradigm shifts in heart failure that have led to new treatments and saving lives every day with these 2 programs in mineralocorticoid receptor antagonism and with beta blockers at Monsanto and at AstraZeneca. In those efforts, Bertram Pitt was part of the first one, and Dr. Faiez Zannad was part of the other one. And this is a picture of our Global Scientific Advisory Board. 2 members from Harvard and Dr. Gunnar Olsson, who was previously global head of clinical research at AstraZeneca. The latest addition is a top opinion leader, Raymond Benza, who's principal investigator for our PH trial in phase 2 in the US. So these are the 3 programs that we are pursuing. And the first one is an HDAC inhibitor. We call it CS1 in phase 2. And we aim to complete the trial and report top-line data by the end of this year. The 2 other programs pursued at University of Michigan is prostacyclin receptor agonist, CS585, halfway through a 2-year program towards an IND submission by next summer. And then we have CS014, that's a novel HDAC inhibitor program. Also that one halfway through a 2-year program towards an IND submission and then into the clinic by the end of next year. Now, why are we in pulmonary arterial hypertension? This is a very severe disease. It affects a lot of people. And what happens is that you have too much pressure in your lungs, you get remodeling of your vessels, and the right heart eventually fails, and these patients die way too early. There is a significant unmet need in this patient population based on both the side effect profiles of current drugs, but also the efficacy needs to be improved. And most importantly, you would like to impact the remodeling aspects of the heart and the vessels and reverse that remodeling. Let's look at that a little further. But first, the market is approaching $10 billion and is growing, and both patients and drugs are growing in sales. Now, there are a number of classes of drugs used today, but basically they are vasodilators, so they lower the pressure, and FDA approves these drugs based on improved physical capacity to walk, 6-minute walk tests. These drugs are not approved by impacting remodeling. Now, you have a strong need for therapies that do that, but also for safer and more tolerable treatments. There's also room to improve when it comes to administration, how patient takes these drugs. So let's go to HDAC inhibition. What is epigenetic modulation? Well, it is epigenetic modulation is how you can impact the expression of proteins that is mapped in the DNA. And in some individuals, these protein expressions are wrong, and you can help them by epigenetic modulation therapies. Cereno Scientific is the first company to deploy this development for clinical use of HDAC inhibitor in cardiovascular disease. The scientific advisory board and the chairman, Dr. Pitt, approached me based on our previous contacts and work together when he discovered what we were up to. And we formed the scientific advisory board that I just showed, and they immediately started to review the data together with us and pursue the publication in The Lancet, one of the most prestigious peer journals on cardiovascular disease and in science in general. And this was published last year. And it discloses the scientific data that's there already that documents in various preclinical models and clinical how large potential there is with HDAC inhibition in cardiovascular disease. And this made Cereno to do a broader strategic review and also reprioritize our first indication, and that became pulmonary arterial hypertension. And there are a number of diseases that we could address with this mode of action. We have chosen pulmonary arterial hypertension because it's a very severe disease, and it fits very well like a glove with the characteristics that our drug has from an efficacy profile point of view. But with all these indications, and in cardiovascular disease in general, there is a disease progression that involves cardiovascular remodeling, both in the vessels and in the heart. And that impacts these patients, and both from a morbidity and mortality point of view. So we have a major potential to impact not only PH, but other indications. It's like a platform for research. Now, this all started with Professor Sverker Jern and his vision that we should have drugs for patients that are at risk for thrombosis development, which is the major cause of death in cardiovascular death, and that impacts major organs such as the brain, called stroke, or myocardial infarction with the heart. And it's an occlusion of the blood's flow that then makes the brain or the heart cells die because of lack of oxygen. The body has its own defense mechanism. It's like sprinklers in a high-rise with water. But in the body, we have not water in those sprinklers. We have a fibrinolytic substance, something that can dissolve a clot that is starting to grow and become dangerous for occlusion. And the body has this defense mechanism to protect itself. But in risk patients, this fibrinolytic substance in this cardiovascular vessel walls is very low. And Professor Sverker Jern identified the ability to upgrade the production, synthesis, storage, and release capacity of that substance with HDAC inhibitor. And that is the foundation of Cereno Scientific. And we have documented that by using such an approach, we can prevent thrombosis development without the risk of bleeding in live animals. So let's move on. We moved towards the PAH indication because that fits the broader scope of HDAC inhibition and the documentation that's there. And this market is really hot. In November, Merck acquired Acceleron for $11.5 billion, mainly because of their PAH program, the most advanced program, which they by hearsay paid $7.5 billion for, a program that had completed phase 2 in the clinic. This is in the field that we are, and the reason Merck acquired this is that there is indication that that drug, sotatercept, has a potential for reverse remodeling, so disease-modifying capacity, not only lowering the pressure. And that's exactly where Cereno is also targeting with HDAC inhibition. Now, let's look at our first and most advanced program, CS1, which has a competitive profile with strong supporting data. Now, back to sotatercept. And here you see to the left characteristics and describing sotatercept. And you see the patent 2036, orphan drug designation, and so on. Potential disease modifying, pulmonary pressure reduction is important and a given. But then reverse remodeling, antifibrotic, anti-inflammatory, anti-thrombotic capacities you would like to have for these patients. And then look at CS1. So these are two different mode of actions. Sotatercept was bought for $7.5 billion in end of phase 2. CS1 is in phase 2. And as you can see, we complement sotatercept. We have the same type of characteristics. And in addition, we are also an anti-thrombotic. And I'll show you some more information and why we and the top opinion leaders in this field believe that we are onto something really important. I won't go into detail here, but this is 4 preclinical studies in rat models in pulmonary arterial hypertension. And there are 2 studies here on sotatercept, and there are 2 studies on Cereno's substance VPA. And you see the different characteristics on the top of this chart: pulmonary pressure reduction, reverse remodeling, antifibrotic, anti-inflammatory, anti-thrombotic. Now, as you can see, has the same kind of efficacy data in all these aspects as sotatercept in preclinical models, in these 4 models. We have a different approach, HDAC inhibition, and they have a monoclonal TGF-beta trap molecule, and they are now in phase 3. And if you look at the market, this is showing on top of HDAC inhibition, there in the middle, and Cereno, are the marketed drugs. They're basically vasodilators. They dilate the vessel, lower the pressure, but they don't claim reverse remodeling or disease-modifying capacity. If you look at below, you see the new developments. These are not on the market yet, and there on the top you see sotatercept, and that's the drug that we are comparing and benchmarking us against. And all of the newer drugs are approaching and trying to prove reverse remodeling. And we believe that we are as good as sotatercept in the preclinic, and maybe the best drug here. We'll see. We'll have to prove it. Now, why is this so interesting? Well, we know from preclinical work that we reduce the pulmonary pressure. We know that we have both prevention and reverse remodeling, reverse the remodeling of the heart and the vessels when treated, that we are an anti-fibrotic, anti-inflammatory, and that we also prevent thrombosis in models, which is an end-stage problem in these patients. And I'll show you here 3 proof of concept studies with HDAC inhibition in cardiovascular disease. This is one of these studies in pulmonary arterial hypertension where we show prevention and reverse remodeling and impact all the inflammation parameters that are risen in these rats when they develop PAH. This is another very interesting proof of concept study. It is, in a way, a proof of concept of SPAF, stroke prevention in atrial fibrillation. That is the major indication for anticoagulants and antiplatelet drugs in the $25 billion prevention of thrombosis market in man. And there's a big problem in that market, as I mentioned up front, that there is too little efficacy and there's a major risk, which is bleeding. So you put every patient at risk of bleeding 24/7 with the current therapies. That's how they work. Now, with HDAC inhibition, we have proven that we prevent thrombosis in preclinic without the risk of bleed. What happens in these patients and why do they get a stroke? Well, there is a fibrotic development, a connective tissue growth in the vessels and in the heart, which leads to supraventricular arrhythmias, and then that leads to thrombosis and stroke in the brain. This is a genetic mice model that develops fibrosis in the vessels and in the heart, leads to supraventricular arrhythmias, leads to thrombosis, emboli in the heart. And if you use our substance compared to control, you prevent mitigate the development of fibrosis, you delay the onset of supraventricular arrhythmias, and you reduce the thrombosis risk and the thrombosis incidence. So this is a proof of concept in prevention of thrombosis in fibrotic animals. This is the third proof of concept in animals, and this is the prevention of thrombosis. You might have read our press release of our new agent this week, that we prevent thrombosis in animal models with HDAC inhibition with CS014. This is a previous study that was done with the substance in CS1. So here you hurt the mouse and they develop a clot, and if you are on HDAC inhibition with our substance in advance, you reduce the thrombosis formation, and that's done by the increased level of the fibrinolytic substance. That's the green here in the picture. So that now I've shown you 3 proof of concept studies in preclinic with HDAC inhibition in cardiovascular disease models. Our clinical program for CS1, our lead compound, has gone through a phase 1 trial, no issues, and a targeted release profile, no issues with bleeding. We are now, as I mentioned, in phase 2, and we're very excited about that study. And so is our principal investigator and top opinion leader, Dr. Raymond Benza, as you can see from this quote. Who is else excited about this? Our global collaborative partner in this study, major healthcare company Abbott. We made a collaboration agreement with Abbott last year that we will collaborate together with them, use their cutting-edge technology to measure pressure in the lungs with an implantable device. It's already approved, that device, CardioMEMS, in heart failure, and pulmonary arterial hypertension patients develop right heart failure. And of course, Abbott would like to move into PAH with their device. So we are collaborating with this in 10 centers in the US. We're very happy about that collaboration. It's the first time in Abbott's history of 120 years that they've made this kind of collaboration with a medical device and a pharma company. Now, the study is in 10 centers. Patients have PAH. They're screened. They're implanted with the implantable CardioMEMS device, stabilized for 6 weeks, and then we have a 12 weeks treatment period, and the patients are randomized to 3 different dosages, and then there's a follow-up period. The key objective of this study is safety and tolerance, but we also measure a number of efficacy parameters, the standard ones, 6-minute walk test and pressure, of course, but also another biomarkers to evaluate HDAC inhibition in these patients and also lead us to advise for the dose for the next study. Our preclinical pipeline is CS585 and CS014, as I mentioned. Collaboration deal signed last year with University of Michigan, 2-year programs for each substance, and we're half year through. And you might have seen that we released the candidate drug nomination for each drug the last 2 weeks. So we're very happy with the development, and we also have some new exciting data, which I will get to. Dr. Mike Hollenstadt is our Director of Translation Research, and he leads our programs at University of Michigan in Ann Arbor. Now, the first program is a prostacyclin receptor agonist. It's very stable, potent, and selective, which is needed in that class of drugs for PAH. So we signed an agreement, an option to get global development and marketing rights by the end of year 2, and we have nominated the candidate drug. We aim to submit an IND submission by the summer next year and move into the clinic phase 1 by the end of next year. The second preclinical program is the HDAC inhibitor. It's a novel HDAC inhibitor that we are very happy with so far, and this is also undergoing evaluation documentation for an IND at University of Michigan. And it has the same target timeline to submit an IND by the summer next year and move into phase 1 by the end of next year. Now, in the last week, we have released 3 new press releases in relation to our 3 development programs. The HDAC inhibitor, no, I should say the CS585, the prostacyclin receptor agonist, was accepted at the European Hematology Association, which held in Vienna in June. And Mike Holinstat will present data. And the title of the talk is CS585 is a first-in-class compound targeting the IP receptor for prevention of thrombosis without increased risk of bleeding. It's the first time this is presented for this compound and for this class. In June 22 to 26, Raymond Benza will present the groundbreaking clinical trial design that we are doing together with Abbott with our lead program in phase 1. He will present that at the Pulmonary Vascular Research Institute in Athens. And the third press release was about our second preclinical program, the novel HDAC inhibitor, which was accepted at European Society of Cardiology, the top cardiology conference in the world per year, and this year is held in Barcelona. There, Mike Holinstat will present a novel HDAC inhibitor regulating platelet activity, fibrinolysis, and clot stability for prevention of thrombosis without increased risk of bleeding. So this is very exciting, and we're making waves in the scientific community. Now, key achievements last year, I've mentioned, we made a deal with Abbott. We made a deal with University of Michigan. We in-licensed CS585, an option agreement with University of Michigan. And we also made a financial milestone, bringing in 95 million Swedish by the early fall. We're off to a good start this year. So we have 2 new patents in our IP portfolio in our 3 patent family, and then we have strengthened our executive management with Fredrik Frick as head of clinical operations, as we soon will have 3 programs in clinic. He comes from Astra, and we have nominated our 2 preclinical compounds, candidate drugs, and then we have also been accepted for 2 presentations for these 2 programs. Now, next milestones are first patient enrolled in our phase 2 trial. There's a slight delay here in relation to getting the ethical approvals and contracting with these centers, but we have initiated recruitment, and we're expecting the first patient enrolled soon. We have a new milestone, a new financing milestone in September, that if fully prescribed, a warrant package will bring in 114 million Swedish. And then, of course, we have our major milestone, top-line data reporting on our phase 2 trial in pulmonary arterial hypertension by the end of the year. So we have a global presence. We are in a profitable market that's growing. We have a very extensive therapeutic potential with our pipeline. We have a strong team. We have a global following on top opinion leaders. And we have external validation from Abbott, Harvard, and otherwheres. So we're very happy with the progress, and I thank you very much for this time.
Thank you very much, Sten, for such a nice presentation. And now we have time for a couple of questions. My first question will be related to your first indication for pulmonary arterial hypertension. You have shown how nicely CS1 regulates the balance in between PI1 and TPA, and TAP, yeah. My question is, one could have expected that you would have gone for a thrombotic disease, but you didn't. You went for pulmonary arterial hypertension. How strong are the vascular remodeling properties compared with the antithrombotic effects of CS1? Would you put it in a balance like 50-50%, or which one is more dominant than another one?
Well, I think, if you look at cardiovascular disease in general, I think the possibility and the potential that's been showed in preclinical models with HDAC inhibition and our substance VPA is very important. And I think that's really the reason why Merck bought sotatercept. But this possibility of impact in disease remodeling applies to many cardiovascular indications. So I think that impact by itself, and as I showed here, the comparison with sotatercept, and I believe that we are at least as good as sotatercept in that these are PH models. On the other hand, the novelty of Sverker Jern's approach with HDAC inhibition for prevention of thrombosis by reducing pi and at the same time increasing the synthesis, storage capacity, and release capacity of the fibrinolytic compound protein TPA from the vessel wall to dissolve locally the thrombosis or the clot as it's building up before it becomes occlusive is truly groundbreaking. And that's the major cause of death on the globe. So in non-communicable diseases. So I think that be able to provide that kind of drug to prevent thrombosis in the patients that are at risk of it will be major. But that's a longer development program. It's more expensive. So orphan drug indications such as PH is the patients that we will help first.
That's very, very interesting. And now moving to your clinical study, you have an important collaboration with Abbott, with the CardioMEMS system. Could you explain what are the benefits of having this device for the study compared with a study without CardioMEMS? And what are you bringing new?
Yeah, so it's a very good question, of course. And what's new, what's good for Cereno here is that we, and for the patients, for the doctors that's studying the patients here, and for the study itself, is that you have a device that's implantable that can measure the pressure whenever you want. Otherwise, you have to bring in the patient and measure the pressure. So you get continuous pressure measurement and you get more exact data. And this has been shown in other areas of heart failure. You don't only get pressure measurements, you get actually some other data about the heart's function. So this will provide us more exact data to evaluate these patients and how they are performing with our drug versus without. And it also is possible to have less patients actually in this kind of study. So more exact and less patients and more data out. The benefit for Abbott, of course, is that they will be able now to study these patients with their CardioMEMS technology. It's the second study that they're doing. And Dr. Benza recommended us to go to Abbott and do this because this is the best way to do the trial. We were on stage with Merck's CMO, with Abbott's CMO, Phil Adamson, and our CMO in Washington with the best people in the world doing clinical trials in cardiovascular disease. And this study was discussed and the way that you do it. So it's a very, very good way of doing a study.
Very interesting. And one final question. Could you remind us how does your cash position look like and your cash runway?
Of course. So we actually today at 8 o'clock released our quarterly report, Q1. And in that it states that we had 77 million in the bank as of the end of the quarter. And we have the possibility, we're fully funded past our next financial milestone, which is the warrant package in September, which we assume will bring in a fair amount of capital to us. The last one was prescribed to 97%. So, but we have the possibility to move to the end of our trial without just a major subscription of that. So we, if fully prescribed, we'll be able to move in and complete our phase 2 trials with the 2 preclinical programs.
Then thank you very much for your nice presentation and for being here today with us.
Thank you, Gonzalez, and thank you, ABG.